Hello, and welcome to Science with Mr. Morgan. In today's episode, we are going to talk about evidence of evolution. First up, we're going to talk about structures. Now, when I say structures, am I referring to buildings and other permanent structures? Nope. Some evidence of evolution revolves around body parts that have lost their intended use through evolution. Now, these structures, the ones that have lost their use through evolution, are known as vestigial structures, or even sometimes vestigial organs. The presence of these structures suggests that an organism changed from using the structure to not using it, or using it for a different purpose than it was originally intended for. Now, some examples of vestigial structures in organisms include, but are not limited to, the following. Ostriches have wings, but they don't use them to fly. Similarly, penguins also have wings, aka their flippers, to swim rather than fly. Whales have pelvic bones to attach to legs, but they don't have legs. This does suggest, though, that they descended from an ancestor that had legs. Humans have a tailbone, but no tail. This part suggests that we once had a tail, at least at some point in time, but we don't anymore. That's enough about those types of structures. Instead, let's look at homologous structures. These are a specific type of structure which have a common function across organisms, and they suggest a common ancestry between organisms. Now, examples of this include the limbs of mammals like bats, lions, whales, and humans that are used to move. They allow us to move around and do things, aka a common function. Since mammals have limbs, it suggests that they all have a common ancestor, including us. Besides similar structures, though, embryos can also provide evidence of evolution. So what is an embryo? An embryo is an animal or plant in its earliest stages of development. And embryology is the study of how organisms develop, aka the study of embryos. Essentially, this is looking at a plant or an animal before it is born or hatched. In this stage of their development, embryos of different species look similar to one another, at least early on. Now, many animal embryos do not differ much in appearance until they develop further, towards the end of the gestational period. That being said, some are also still very hard to tell apart, and there can be unexpected traits that appear in animal embryos. But what do I mean by unexpected traits? I mean that embryos can present traits that are not seen in the organisms after it has passed through its gestational period and is born. For example, all vertebrate embryos, like us, have gill slits which are not actual gills and do not typically last past embryological development. But if they don't last, then what is the purpose of them? Specifically with gills, they connect the throat to the outside early in development, but eventually close in many species. Even though they close in many, these gills do remain present in fish and larval amphibians as full gills when they're finished developing, because they use them. They have a need for them. In mammals, however, these gill slits form part of the lower jaw and the bones of the inner ear. Now, these similarities between embryos suggests common ancestors, again, amongst mammals in this particular example. Another typical example of common ancestors that you're probably familiar with is humans and chimpanzees. Now, it's important to note that humans did not evolve directly from chimpanzees, but instead the similarities between the embryos of both species suggests that humans have an ancestor in common with chimpanzees. As our common ancestor evolved, humans and chimpanzees went down different evolutionary paths and developed different traits. Now, besides similarities in structures and embryos, molecular evidence also supports evolution. Some of the best evidence of evolution comes from examining molecules and DNA found in all living things. Since the 1940s, scientists have studied molecules and DNA to confirm conclusions about evolution drawn from other forms of evidence. One particular thing that is used to study DNA in relation to evolution is called a molecular clock. Molecular clocks are a device that are used to determine how closely two species are related by calculating the number of differences between the species' DNA sequences or amino acid sequences, and then comparing them. Sometimes molecular clocks are called gene clocks or evolutionary clocks as well. With these, the fewer the differences in the DNA, the less time since the species split from each other and began to evolve into different species. Now, looking at specifics, let's talk about chickens, gorillas, and orangutans. Chickens and gorillas have more differences between their DNA and amino acid sequences than gorillas and orangutans do. Since chickens and gorillas have more differences in their DNA and amino acid sequences, This suggests that chickens and gorillas had a common ancestor a long time ago. They're not closely related. That being said, since gorillas and orangutans have less differences in their DNA and amino acid sequences, this suggests that gorillas and orangutans have a more recent common ancestor, aka they're more closely related. Summarizing that part of what I just said, this information suggests that the gorilla and orangutans are more closely related than the gorillas are to chickens because of those differences. Now, it is important to note that animals may look similar, but they can have very, very different DNA sequences and evolutionary ancestry, which means that they aren't related as much as they appear to be. Remember, genotype versus phenotype. Now, another scientific development that provides evidence is the sequencing of genomes. What's a genome? It's the DNA sequences of all of the genes of an organism. And so far, many have been fully determined for many different organisms. We can use these to compare genomes to provide new information about the relationships among species and how evolution occurs. And that brings us to the end of today's lesson. Until next time.